Okay, the light reactions, what happened there? The light reaction, of course, requires sunlight to go. We know that because that's when it's split in the water. And the ATP synthase is used to, somebody walk. There it is. Here, Roger, here's the bacon. Goes back and you wouldn't hear. We know that whatever is produced with the light reactions goes to Calvin cycle, which is the dark reactions. And I showed you a picture of the light reactions showing you the two photo systems and how we had named them wrong because we discovered them in the wrong order, basically. Okay, so this one happens first, of course, and then that one happens, but that's when we call one code. We discovered this one first. And then we began to wonder about all the electrons leaving it. Where are the electrons coming back from? Then we go, oh, there's another one that's providing electrons, so that worked out pretty well. And there's the ATP synthase. Right here, it looks like I've seen it portrayed all kinds of ways, sometimes like a funnel, sometimes like a windmill. There's no particular look to it. It's just the fact that hydrogen makes it go, and the hydrogen passing back from the inside the thylakoid to the stroma is what produces the ATP. And in the stroma is where the next cycle is going to happen. And there's the summary where this one happens first, so it's called number 2. And this one happens second, so it's called number 1. And everything then moves into the dark reactions. So now we're on the dark reactions. We're going to take it slow again and work it slowly so you get sense to it. Okay. This is where the carbon gets fixed and makes glucose. Like I say, here, if you're going to put, like, you're going to put a mailbox up and you want to put the post in the ground and pour the cement, right? You want to fix that post first, don't you? So you put the post in the ground, you pour in the cement, and then the post is fixed. Well, all this, you got to fix that carbon in order to make the glucose, which you want. Actually, photosynthesis is not about making oxygen. By now you should know that. It's about making glucose. Oxygen is an offshoot. Die for it, but it's not what is happening. And the carbon and the oxygen that's in the sugar is coming from the carbon dioxide. The hydrogen part is coming from this, which came from the water, remember? That was split in the first one. So all 3 members are here. They're going to make this. 2 of the members will enter the dark reactions, and 1 of the members is grabbed by the NADP and carried to the dark reactions from the light reactions. All right. These dark reactions don't happen in the dark. They just don't need the light because their energy supply comes from ATP, which is made by the light reactions. They need light. But just because they're called dark reactions, don't think they happen at night. They do not. Well, they do. But the main thing is that they have no dependency on light to make them go. Because their energy is coming from not the sun, but the ATP. Which, of course, came from the sun in light reactions. So all you need now, when you got the H's coming in, from the light reactions, and you just need the carbon dioxide to provide the other 2 components. So here's how it works. Okay. Here's the carbon dioxide. Okay. This RUBP is cycling around. It's cycling around. That's part of the Calvin cycle. You're going to find out that this 1 carbon slides in right there. So that now you have this makeup. 1, 2, 3, 6. There's your 6 carbons that you're going to need to make your 6 carbon glucose. The thing about it is, photosynthesis doesn't really make glucose, it makes 2 of these. If you look, they're identical, but they're flopped over. Both of these are 3-PG, phosphoglyceric acid. And this one is these 3. And this one is those 2. When that bond is broken, this swings around and ends up right there. Everything else is already here. You see your phosphorus right there? That phosphorus is the part of the name called phosphate. And that's going to have a need later on. I'll show you a cycle in a minute. But the thing about it is that this carbon slides in right there. Actually attaches there, and there it is with a double bond. And this is the first thing you make, which immediately breaks in half. And like this, these 3 go on to produce this 3-PG. And that swings down here where the bond's gone and makes, and actually they're identical. I mean, I got this, I got this, I got this, I got this, and now I got this with the 3. So these 2 are, I mean, they come together, you'll have your 6 carbon glucose. But that plant can use this to make all kinds of stuff more than glucose, like cell structures, leaf tissues, things like that. Okay? This is Melvin Calvin. He's the one who the cycle, he's the one who figured out what was going on. And he's a modern, he's a scientist of our day. We're not talking about somebody who lived 300 years ago. So the Calvin cycle is, the cycle has been around a long time, but the name Calvin has just been addressed to it. He was the one who said take 6 molecules of carbon dioxide to make just 1 molecule of glucose. Because I need 6 carbons. Well, in 6 carbon dioxides, I got my 6 carbons. I got 12 oxygens, though. But I do have my 6 carbons. So he was the one who realized it's going, for every glucose you manufacture, you've got to use 6 carbons to make it happen. And when he said that, then we began to figure all kinds of stuff out. And that's why he didn't name it himself. Scientists named it after Calvin. I guess no one did more than Calvin to help us understand the cycle that happens in the dark reactions. I looked through all kinds of diagrams to find the one I thought I could most easily teach. And then I put it in parts like I did last time. And this is the one I'm going to use. This is the one, and I'm going to, as you see, I'm going to break it down. But right now, this is the overall view. See that that ribulose phosphate coming around I tell you about? 1, 2, 3, 4, and 5 of these. Now go back and look. Remember right there I tell you that he's coming up. He's coming up. And he's coming up to join the carbon dioxide. Well, there he comes. He's coming up. And there's your 6 coming in. Well, 6 and 6 equals what? That's why I have 12 of these. But when you take, if I have 6 of these 6 carboners, I can cut them in half and have 12. Still 36 carbons, right? I mean, what is 6 times 6? 36. 36. Well, what is 12 times 3? You still got your 36. It's just they've been torn in half. You know. Now, a number of things are going to happen. And remember, I said that these things come off. This is what you're trying to build. But if you look at them down here, there they come. There they come. They're coming off. And then these can be used to manufacture. And notice, I call them 3PG. Here they're called G3P. Just same thing, different name, same thing. And as it moves around, you're going to see things happen. And these 10 molecules will break down to become 6 of those. And when I do it one by one, I hope it make more sense. Because I looked at a lot of diagrams where I said, that's the one I want to teach. And you learn this, go back and look at the other ones, see if you can follow them. I can follow all of them. I ain't going to teach those. They're too hard to follow. So here we go. Now what I'm doing, I'm taking just this part. I'm going to show you just the top part. All right? I already told you this. Carbon dioxide, 6 of them. Because I need 1 per of these, and I got 6 of these. So the carbon dioxide enters the Calvin cycle, and when these come together, I will have 6. But notice 1, 2, 3. Now there's 6 carbons and not 5. Because this 1 carbon bumped it up to 1. It's really unstable. It won't stay. It'll break in half, just like raw spaghetti breaks in half. And there's no name for it that I ever saw. I don't know why they have never named it, but it just going in flash. It's going to break in half. So now I got a P and 3 of these, and a P and 3 of those, and we're going to see that happen on the next thing. So if I have 6 these and I cut it in half, I have 12 one of them. So if I cut it in half, my 6 become 12 of the halves. Now, what I'm showing you here is that this has come around, you produce this, and now I'm skipping this, and I'm going right to here. Because this don't stay very long, but it's there for a minute, and then this immediately becomes that. And notice how 6, when you have it, you'll have 12 of the 3s. Now, 6 times 6 is 36. 12 times 3 is still, they're all there. They're just in half. Now, something happens here, coming around. Now, there we are up there. Now, ATP provides energy. So what happens here? NADP, H, provides the H. And what happens when you drop off these P's, they come out by themselves. But the fact of the matter is, you've now included H in your compound. There's H in here now. And the energy from here provided the power to put it in there. This is your G3P. Okay? Now it's going to come on around. Now, there's this one now again. Come on around now. I had 12, right? I lose 2 of them. How many are left? 10. So I had 12. 2 of them were spit out as the product. 10 of them going around. Now, I've still got all my carbons. It's just that now I've lost 6 carbons in these compounds. Well, what is 36 minus 6?
30.
You're back to 30. So you see, your 30 carbons is because you just spit out a total of 6 of them. There's 3 in these, there's 2 of each. Now, remember that one going up has 5 carbons? Remember he has 5 carbons? Well, here's 10 of them having 30 carbons. If I had 30 carbons, how many of those 5 carbon compounds can I make if I have 30 carbons?
6.
I can make 6 of them. And remember how 6 entered the picture. So I'm taking these, now right here now, to just reshuffle. 6 times 5 equals what? 30. I got 10 times 3. These 30 are reshuffled to give me 5 of these RP compounds. And that's going to come one more step and produce that product, RUBP, that enter the process. It keeps on going. And every turn spits out what can be 1 glucose. Now I told you earlier, it took 6 carbons to spit out that 1 glucose. Because the other 30 carbons are used to rejuvenate this cycle to do it again. As long as you provide the carbon dioxide and the ATP and the hydrogens, this sucker is going to run forever. So now we're right here on this one. And like I said, I have 30 carbons in these 10 compounds. Hocus pocus, abracadabra, and these 10 compounds become 6 compounds with 5 each. I still got my 30 carbons. So it's just being reshuffled now to make 5 compounds. And so just right here, went the wrong way. Okay. See that right there we talked about? Now, if you notice, there's a P on here. And when we started, there's another P. There's another P. And what happens is when this thing rolls up, ATP comes in, we use the energy to attach this phosphorus. This phosphorus came from here. And now this is right back where we started, so we can do it again. And let me go back to my first one and I'll show you again on the overall one. Okay, here's that now. Again, this is the start-off compound for the Calvin cycle. RUBP has 1, 2, 3, 4, 5 carbons. When it joins this carbon, now you have, you're going to have 6 carboners, which split in half to form 12 3-carboners. And there's one right here I didn't put in because remember it goes so it's not there very long. Remember that one right there I said it's not there very long? This one is not in my picture. I might one day go back and put it in there. But each one of these carbons is added, and I have 6 of these unstable 6-carboners, but they immediately split in half and form 12 of these. I think I will go back later on and take this one and insert it. Just to let them know that, well, these two do produce this one, which is not there for a fraction of a second, because this one immediately cuts in half and forms 12 of the 3-carboners. So that's where this 12 came from. So 5 plus 1 gives you 6, which immediately break in half to form 12 3-carbon compounds, PGA. And then when you tack on energy, and notice how this phosphorus doesn't go up there, it stays by itself. The energy is used to put the H in the compound. And I need, if I have 12 of these compounds, I need 12 of the H's. And that's this one and this one. The only difference is he does not have the hydrogen, he does. And now I have 12 of these, 2 of them come off and can be joined to make a single 6-carbon compound. But 10 of them going around and get reshuffled to this. In comes ATP to provide the energy to attach the phosphate, because see, when I cut it in half, I do have 2 phosphates, don't I? I can't have 1 phosphate, because the phosphate is important to make this thing go. And there are other ones I found. And now if you see this, I go back and try to find them, see if you can follow them. And I might one day put a few more on my slideshow and y'all, let's follow it through another picture. I do have this though. That thing right there. That's just a summary of everything at one time. That's what's happening inside it. This is a chloroplast.
Okay.
That's the chloroplast. The green looking things are the thylakoids. Something happens in the thylakoids, and this out here is the stroma. Well, the light reactions where the water is split, oxygen is given off. The H, see how Na comes in, it leaves carrying the H with it, and then it goes and enters the cycle. And you see how the ADP and the phosphorus come in, soak up the energy and make ATP. That's going to drive this cycle. And then you see here that in this cycle, the RUBP, that 5-carbon compound, is going to join with carbon dioxide, but make a 6-carbon compound that immediately breaks in half, remember. It forms 2 of these PGAs, phosphoglyceraldehyde, and then it is reshuffled by adding hydrogen, which is right here. They're not trying to show you the actual, because this part here should be coming over here. These 2 parts should be entering right there. Because I need this hydrogen to become available. I need the energy to make it add here. Because when you add the hydrogen, this becomes this. Now, I told you earlier, you had just made 12 of these things. With 10 of them going around, 2 of them are spit out, and they produce the sugar that's exported, or that you eat yourself when you eat the vegetables. And that's photosynthesis. Now, I don't know, which one do you think harder, respiration or this one? To me, they're not, and I don't expect you to remember these big old fancy names. I do expect you to remember that the compound that meets that incoming carbon is a 5-carbon compound. Can't be 6 already, can it? That'd be too many. Because everything's based on 3. So if 5 comes up, this is a 5-carbon compound comes up, it's going to carbon dioxide, 5 plus 1 is 6. But that 6-carbon compound immediately just breaks in half to form 2 of these things, which when you get the hydrogen, they're ready to be spit out and make sugar. And I mean, if you're making 12 of them, most of them stay in the cycle. Only 2 of them come out, so the majority of them never leave the Calvin cycle. And so, for you to make 6 of these things, I got to put in 6 of these things, because there are 6 carbons in that sugar molecule. And this one is not trying to show you the 6. I picked that because I like the numbers. I don't want to show carbon going in. I want to show 6 carbons going in, meaning 6 of these coming up. That would give me 6 of these unstable that I don't have on here, which immediately break up into 12 3-carbon compounds, which is just this one. And this one, all he needs is the hydrogen, and he can become that. But you need the energy to make the hydrogen bond with him, so the energy is provided. The NADP gives the hydrogen. He's back plus because he gave off his partner. He's going to go back to the light reactions and pick up some more and bring back. These are going back to the light reactions to be readjusted and bring back some energy. So these are used up. These are to be used. So these 12 3-carboners with these 12 and these 12 take on hydrogen and become this. Okay? And I have 12 of them, because I had 12 of these, and I have 12 of those. Now these 12, like I showed you, 2 of them peel off and can be used, they can put them together and make glucose. The other 10 are then reshuffled, because I need a 5-carbon compound. I don't need 10 3-carboners, I need 5, so they just reshuffle, using energy, of course, to do that. And when they reshuffle, I'm right back to where I was, so I can do it again.
Why
And
does it lose two right there?
lose two where? What are you talking about?
The two molecules, why does that happen?
Because the whole thing is designed to make glucose. Well, how big is glucose?
36?
No.
Wait,
One molecule of glucose is C6H12O6.
oh,
That's 6 carbons. How many do you need to make one of those? I need 2 of the 3 carbon compounds. I mean, 6 of these only spit out 1 glucose.
so it's still a 12-3 carbon, but it becomes a sugar.
Well,
12
these
carbon
12
with hydrogen,
entities,
right?
the 12 entities, any 2 of them will work. But just 2 of them is all you need. Because 3 plus 3 makes
But
the
only
6
thing-
carbon
The only thing
glucose.
that changes from here to here is it becomes a sugar 3 carbon with hydrogen on it, and
Yeah.
then it drops off too?
Yeah.
Okay.
And all 12 of these look alike. They're all identical. Just the 2 of them are spit out.
Well, what makes that happen right there?
Enzymes.
Okay.
Enzymes make everything happen. I didn't name the enzymes, that's a whole other ball of wax. But it all goes, enzymes make it all go. Enzymes make everything go. This is metabolism. Now would this be anabolism or catabolism? What'd you call this?
It's beauty.
Because you're making something.
Yes.
That's anabolism. I know you're tearing things apart and you put things together, but the overall picture, you're making something. Now I know you gotta, even when you make a cake, you gotta do some destruction to make a cake. So, the idea of this 3 splitting to make this one, that'd be kind of catabolic. But that's not the whole picture. The picture is, I want to make something. If I got to tear something up and make something, I will. Now, respiration, what would you call that something? Would that be anabolic or catabolic?
Respiration.
So what are you doing to glucose to get out the energy?
Breaking it
You're
down.
tearing it all to pieces. So that'd be catabolic. And you got to have it. So you got to tear it up to get energy out. And now, when you breathe out the carbon dioxide that came from that food, guess who can use it again then?
The plant.
The plant. And when he breathes out the oxygen, I say he breathes, when he releases it, well, you can use it. So the carbon dioxide has been here for eons. You never use it up. It just recycle. There's nothing gained, nothing lost. If you put plants in here and seal things up and they could grow, we could live here with these plants all the time, eat just plants. But if you had one banana plant and 13,000 people, that ain't work. And we're scared that what happened to Earth, we're cutting down all these trees. That's like having one banana plant and 13,000 people. You won't destroy the Earth, but you might destroy the Earth's ability to sustain population. What good is the Earth to me if I can't live here and grow old and die? But I learned it a long time ago, and I relearned it, and I wish I knew back then what I know now. I'd make straight A's in biology.
Go back to the picture real quick.
That one? This is the one. I monkey with things so much. I'm not sure if that's the actual one I found or one I created from one I found. I know this all here was not there. I put all this in myself. I took these arrows and I flipped them, flopped them, and stuck them on there. If you look real carefully, you see where they're not always exactly lined up, but I do pretty good with it. So I don't know if you're going to find one. You'll find one that you'll know that's what he used because you recognize these colors. But you'll be going, oh, he did something here and he did something there. But I think I will go back and do my magic. And that one I showed you right there, that's gone real fast, like a flash of a pan. I might put him back in here, right up here, just to show people that he is there for that long. And he's, separates immediately. But I don't know where I got that from. I may have made that one myself. I don't know where I got it from. So chances are, I took 2 of these and then flip one over and change the P and stuck it on there and created that thing there so I could tell you that he is produced. But most books don't show him because he is not there but for a split second. But I thought they'd show him that step. That this thing does exist just for the tiniest fraction of a moment. And then he immediately splits and becomes those. Okay, so what we have here now, one more time, let you know this. Here are the light reactions having two photosystems. Okay, they both have transport chains. They use different wavelengths. This one uses a 680 wavelength. He uses 700 wavelength. Same color basically as you're looking at. And it's not green, or green's been reflected. That's the reddish wavelength. Now, so you water your plant, roots, not the leaf. The xylem drains the water up to the leaves, out to the veins to the cells, and the cell of the chloroplast. The water is the chloroplast. The light from the sun strikes the thylakoid, producing high energy. That splits the water. The oxygen, it doesn't need the oxygen. It gets rid of it. I need that. But the hydrogen has got to be saved to be used over here. Now he does do something. He says, oh, as long as I got it, I want to have it pass through here and make some energy. He does use the hydrogen because hydrogen's got to get out there. Remember when I showed the other pictures to you, the hydrogen is inside for the longest time. See how the hydrogen is out here? Well, this is not where the Calvin cycle is. That's inside that green thing. The Calvin cycle is outside here. So the water is being split inside, and I got to get these outside, and the body figured, well, since you're going outside there, go down through here and make me some energy. You know, if you go into the store, while you're there, pick me up some milk. Save me a trip. So since you got to get in here anyway, just go and pass on through here, and as you go back in, make me some ATP. And nothing's wasted. And now, this is outside the thylakoid. I'm showing you a picture now. Well, go back where I was. Everything happens inside here. And when the hydrogen passes through the structure, it goes out here. And it's immediately snatched up by NADP because it would leave with this is a gas, right? It would leave with the oxygen. You don't want the H leaving because there are airways out here, and that H would leave right along with the oxygen. That would not be good because you need the H to make the glucose. So the NADP said, you stand here. It grabs it and forms NADPH, which then carries H to the Calvin cycle, and that H can then be used to manufacture that G3P, which is then used to manufacture glucose and other plant products. It's a really unique thing. And like I said, the guy who first figured out the really intricate work was Calvin. And that's why we call it Calvin cycle. I don't know any names of people for this over here. It's just light and dark reactions that we know of. But that's pretty cool. Now we're going to do this test the next time I see you, which will not be tomorrow, of course. I won't give you the time to study for it. So next Wednesday, I will give you the test on the photosynthesis lesson. But right now, I want to start my next lesson